Good afternoon, everyone. Thank you for attending this session. I'm going to introduce you to AskBio. AskBio is a company that's been around for a while. We were founded in 2001 based on the work of Richard Jude Samolsky. Jude was the first to clone AAV for therapeutic purposes and is widely accepted as the founder of the field. In fact, he taught Kathy High and Jim Wilson how to make AAV. Many years ago, parents approached Jude because they had children with devastating disease for which medicine had not provided any solutions. And they worked with Jude to try to advance therapeutics to help their children. These children suffered from Duchenne's muscular dystrophy, Friedreich's ataxia, and giant axon neuropathy. And that was the basis for AskBio. We were funded initially by the Muscular Dystrophy Association. During the last 20 years, we were joined by many of the leading AAV scientists, including Chris Bankowitz, Philippe Mollier, Nathalie Cartier. And so now we feel like we have the powerhouse of AAV pioneers and experts in the field. Our mission is to transform medicine, increase access, and lower costs. Over the last 20 years, we've done a lot of different things. Hang on. It's a little tricky there. We are actually generating quite a bit of revenue. We generate revenue through licensing fees and through manufacturing. Last year was the first year where we actually weren't, net income positive. And as a consequence, we just raised our Series A financing this spring. We raised $235 million. We have a fully integrated gene therapy platform, and we have proven scalable manufacturing capabilities. This has allowed us to develop a robust therapeutic pipeline. We have over 500 patents, and we have over 150 professionals. We're currently operating in 3 countries. Now, some of you may be wondering and asking yourselves, why is AskBio presenting instead of Synpronics? Because Synpronics is supposed to have this slot. And the reason is we bought them about 8 weeks ago, so they're now part of the AskBio family. From 2001 to 2016, for those of us who have been in the field, you know that there was a dearth of funding coming into this space. And AskBio decided that rather than take significant dilution, in fact, the first term sheet we got was a $1 million investment on a $1 million pre-money valuation, and it doesn't take a genius to figure out that's not a good deal. So what we decided to do was to take pieces of the platform technology and spin it out into what we call special purpose entities. These were corporate entities that would focus on utilizing the platform for certain therapeutic applications. Nanocor was one such example of these SPEs. Nanocor is working on the development of heart treatments with Medtronic support. Bamboo Therapeutics was the second SPE that we formed. Bamboo was focused on the treatment of Duchenne's muscular dystrophy. We sold that to Pfizer in 2016. And the third one was Chatham Therapeutics. Chatham focused on the use of the platform for the advancement of therapeutics in hemophilia. We sold that in 2013 to Baxter, which is now Takeda. And Takeda recently advanced our program for hemophilia A into the clinic. I like to put this slide in for all of the venture capitalists who said that Duchenne's muscular dystrophy could not be treated by AAV technology. You see Connor, who entered our trial, which is now being run by Bamboo Pfizer. In order to get into the trial, he had to walk up 4 steps, which he had difficulty doing. 6 months later, you see him going up the Durham Bulls Stadium steps with no problem. Gene therapy does work. When we talk about our integrated platform, what does that mean? Well, we think you have to have 3 buckets of capabilities in order to successfully develop AAV gene therapy therapeutics. First one is you need to have sufficient technology. And we have a very deep, proven toolbox that can address a wide variety of tissues. First of all, we take the naturally derived serotypes and we re-engineer the capsids in order to target particular tissues, detarget other tissues, minimize the effects of neutralizing antibodies. We also look at promoters, and now with Synpronics being part of the family, we optimize promoters to control transgene expression. We also look at the regulatory sequences. We are concerned about repeat dosing and have several strategies. We expect to put one of those strategies in the clinic next year. And we're also looking at inducible promoters to turn on and off transgene expression. Manufacturing is something that's very important in this industry and still plays. A lot of our colleagues, we have a HEC-293 cell suspension system. We have high yields, 10 to the 17, with few empty particles. It's universal for all serotypes. It's scalable up to 2,000 liters. We have GMP certified facilities. Our cell line is used by 2 big pharma. We backward integrated because plasmaless supply was one of the big rate limiting steps, and we forward integrated because we want our therapeutics to be stable and have a long shelf life. Clinical translation is important. Again, we have many of the AAV pioneers and leaders. Collectively, we've translated over 12 drugs into the clinic. We have a proven track record. We have proprietary tools to assess which therapeutics are ideal for AAV and which maybe are not so ideal. We have proprietary animal models. We screen using IPS and organoids. We use machine learning to optimize our promoters and our capsids, and we, as a consequence, have a robust pipeline. Because we have an extensive toolbox of capsids and promoters, meaning we're not just in-licensing a capsid for particular use, but actually we have lots of capsids and lots of promoters. We also think that all of the animal models are not very predictive of what happens in humans. The immune system is very complex. And so as a consequence, our strategy is to go in the clinic, see what happens, and make modifications. We can do that because of the fact we have integrated manufacturing. We have captive capacity. Our system is universal. It can produce all the different serotypes and chimerics. And we have an abbreviated setup time. Whenever we are designing our capsid, we always use self-complementary vectors. There's 3 benefits of them. 1, transgene expression turns on quicker. 2, you need less virus for therapeutic dose. And 3, we have found that they're just much more effective and titratable in addition. So when we're designing our capsids, how do we do it? There's 3 steps. Number 1, we look at the target cells and the desired distribution within those cells. We look at the route of administration. Then once we make capsids, we think about NAVs. What is the cross-reactivities with pre-existing neutralizing antibodies? We look at payload size. Manufacturability is really key. If you have a great capsid, but you can't manufacture it in high amounts, it's not very useful. And we look at the IP situation. We have 2 ways today of evolving capsids. 1, we use directed evolution, where we fragment the DNA of the different natural serotypes. We reanneal them into chimerics, screen them through relevant animal models to see the tropisms of the different capsids. We also use rational design. We use the crystal structure, mass spec, bioinformatics, machine learning to intelligently design capsids. And then we have to characterize our capsids. We do it both through in vitro and in vivo. Usually in vivo is preferred methods. We use organoids, iPS cells, et cetera. But there is no perfect predictor for humans. Here are some examples of how we have modified the natural serotypes. On the left-hand side, as we all know, AAV9 goes into the liver in high amounts. What we did is we modified the capsid such that it would have high transduction in muscle and skeletal tissues and in heart, but it would detarget the liver. Another example in the brain is what we call AAV2G9. The receptor for AAV2 is heparin. The receptor for AAV9 is galactose. We took the backbone of AAV2, we stitched in the receptor from AAV9, and as a consequence, we had a capsid with 2 different receptors. So what does that mean? Why is that beneficial? AAV9, of course, it crosses the blood-brain barrier, but it goes into astrocytes as well as neurons. Our AAV2G9 only is neuronal, and there are certain diseases where you do not want to go into astrocytes. Also, the problem with AAV9 is while it can cross the blood-brain barrier, it can also leak out of the brain, and high amounts go into the liver. As you see with our 2G9, we've detargeted the liver, so there's less virus ending up there. But the capsid is something that a lot of people in this space talk about, but they forget about the rest of the regulatory sequences. There's also the promoter, and the promoter controls transgene expression. There's the 5' UTR. There's introns. Of course, the transgene, we've been codon optimizing transgenes for a while. And there's the 3' UTR. Oh, and there's some really neat things you can do with the ITRs. So we focus on all the different pieces. And again, Synpronics has been added to the family to focus specifically on the regulatory sequences. Manufacturing is key. We hear a lot about this. When I used to talk about manufacturing 5 years ago, nobody understood why we spent so much time on it, and they were just like, get into the clinic. Why are you wasting time on manufacturing? I think now everybody understands that the process is the product. And so we invested in a cell suspension HEC293 system. It is relative to the Bacilla virus or the herpes system. It's much faster, it has fewer steps. Our system we call the Pro-10. It's used by Pfizer as part of, they got access as part of the Bamboo acquisition, and by Takeda. We have done over 12 clinical grade productions, thousands of research grade. It has been scaled to 2,000 liters, has 10 to the 17 yields, few empty capsids. We have also invested in plasmid. We are now using doggy bone, well, we'll be officially using for our clinical programs, doggy bone DNA. The benefit of this system is it's not made in bioreactors with E. coli. It's made with a biocatalyst in a test tube. It is a much quicker process and a much cheaper process with fewer bacterial contaminants. We are focused initially, like most of our peers, on monogenetic diseases. It's relatively de-risked there because we know the drug. The drug is the good gene that's going to replace the defective one. We are also making some inroads to the larger indications, to pathway diseases. And again, things like inducible promoters are important for those applications. I'm not going to talk a lot about our pipelines. One of the benefits of being a private company, we are in the clinic for Pompe. We will be in the clinic for limb-girdle 2I and heart failure within the next several months. We will also be in the clinic in the next 18 months for an undisclosed therapeutic. We are working with Chris Bankowitz in his Parkinson's program, providing support to his efforts there. So this is a good summary slide, I think, about how we differentiate ourselves from the rest of the industry. Most of the companies in this space are still using adherent systems for production. They have not really focused on the promoter, and they are still using DNA plasmid as their starting material. Very few players are thinking about the storage needs for AAV, which is quite complex, negative 80 degrees. We thought about for our Duchenne's program, it would take football fields in order to hold all of the different freezers, right, for our therapeutic. We are focused, most of the players are not focused on repeat administration, and they rely on in-licensing of capsids and in-licensing of programs. In contrast, AskBio has the Pro-10 HEC293 cell suspension system, which has been scaled up and has proven to be very reliable. It has focused on synthetic engineered promoters to enhance our therapeutics and to create inducible promoters for turning on and off transgene expression. We have backward integrated into doggy bone DNA, essentially to reduce a lot of the obstacles to efficient delivery and to reduce costs. We have forward integrated into the thin film technology, which allows us to store the virus in almost like a Listerine film. And so instead of having freezers holding lots of vials, we now will have reams of thin film holding our AAV. We have focused on repeat administration and have several strategies that we'll be advancing in the clinic. And because we have a very big toolbox, we're not reliant on in-licensing capsids or promoters from anybody. We basically have the tools necessary to advance a lot of different therapeutics. We are recruiting, so if anybody's interested, that's my email address. It's a very competitive market, so I can afford to be a little ugly about this. So thank you very much for your time.